Okay, today we are honored to have Dr. Bibhaskar with us, who is consultant genetics and molecular medicine at Kokilaben Dhirubhai Ambani Hospital, Mumbai. He is skilled in clinical research, medical education, life sciences, oncology, and medicine. And he has done PhD Life Sciences Genetics from Utkal University. So we are here to discuss about incorporating NIPT, that is non-invasive prenatal testing, into the fabric of precision medicine. Welcome, Dr. Bibhaskar.
Thank you, Dr. Manisha, and thank you very much for giving me this opportunity so that we can discuss about the really a very important aspect of technique, important technique. Now it is very much important in prenatal genetic testing.
Exactly.
So
So my first question is, could you share
yeah.
with us some information on what is NIPT and how does it differ from traditional prenatal screening methods?
Uh, NIPT, I prefer to call this technique as NIPS, non-invasive prenatal screening, rather than NIPT,
Yeah.
because this is a screening technique or method,
Yeah.
and it has been actually the terminology of NIPS has been actually given by American College of Medical Genetics and Genomics. It is a method of determining the risk that the fetus will be born with certain genetic abnormalities, and we are actually uh, uh, only giving importance to the aneuploid screening mostly, because the genetic burden in India is quite high, because 1 in 700 children are born with Downs all over the globe.
Yeah.
But in India, if you look at the incidence, 1 in 1,250 or 1,150, that is according to Dr. I.C. Verma, one paper I was reading, recent paper.
Yeah.
This is the actually risk.
Yeah.
So therefore, these particular things, 34 children are born with Down syndrome every year.
Oh my God.
So the kind of burden that we have is very huge. In other way, if you look at the Edwards syndrome and Patau syndrome, it is 1 in 5,000.
Yeah.
So this test analyzes this particular NIPS, non-invasive prenatal screening. It analyzes small fragment of cell-free DNA, fetal DNA, that are circulating in pregnant women's blood.
Correct.
So this NIPS is performed through two commercially validated methods. One is next generation sequencing. The other one is the SNP. So next generation sequencing involves counting DNA sequences. That is across the entire genome. Whereas the SNP targets specific segment known as SNP, that is single nucleotide polymorphism. So this process actually involves extracting DNA, fetal DNA, and amplifying it, aligning the sequences, and then identify the origin of each chromosome through bioinformatics analysis to interpret the results. So this was actually introduced for clinical use or practice way back in 2011 in USA, and later on, late 2011 or 2012, it is established and popularly, being used by our physicians and most of the laboratories. And in Australia, if you look at, they actually came later in 2013.
Okay.
So I just wanted to highlight here what is cell-free DNA. Is an extra cellular or extremely fragmented DNA, around 200 base pairs, produced during the process of cell death. We call it apoptosis.
Correct.
And mother blood contain her cell-free DNA, as well as the cell-free fetal DNA derived from placental tissue. It is a very beautiful thing which was developed way back in 1997 by Dr. I was just reading one paper, very interesting paper by Dr. Dennis and Lowe and his team.
Okay.
So that is how we could able to utilize this, because the fetal cell-free DNA could give lot of genetic information.
Correct.
That is how this technique developed.
Right. So, okay.
So yeah, please go ahead.
So I would definitely like to ask you that what interventions can be taken, the information which you get.
I am saying this non-invasive screening,
Yes.
when I'm talking,
Yeah.
and when I talk about invasive screening,
Yeah.
is completely different.
Yes.
Non-invasive means it is maternal serum screening.
Yes.
So there are triple screening, quadruple screening, where you have alpha fetoprotein maternal. Then now these are not the confirmatory test.
Yeah.
These are non-invasive testing, but these are not confirmatory test.
Yeah.
The confirmatory test is only through amniocentesis and chorionic villus sampling.
Yeah. That's what
So that is the guideline. It is very clear. That is the guideline given by American College of Medical Genetics and Genomics and American College of Obstetric Gynecology. So this is a foolproof, it's a guidelines which we are following all across the world.
Correct. So that's what I got to know. Ki NIPT is a screening test, and we have got the confirmatory test to confirm whatever information we get from NIPT. Right.
Yes.
So that's great. So could you just tell me, like, what can be the factors that contribute to the possibility of false positive or false negative results in NIPT?
See, false negative result in NIPS,
Yes.
I will call NIPS,
Okay.
occurs when the test fails to detect an existing chromosomal abnormality in the fetus.
Yes.
So the NIPS, when the test fails to detect an existing chromosomal abnormality in the fetus, so
yes.
then only we'll call it a false negative.
Yes.
And this can happen due to low fraction, fetal fraction.
Yes.
And I will add here one very interesting aspect.
Yeah.
The basal metabolic index, mostly seen in our Asian women, or with special reference to Indian context, it is much lower compared to West.
Yes.
So the false negative are higher percentage in Western countries than our India. And one of the paper of Dr. I.C. Verma, when I was going through, he is the father of medical genetics, and a lot of work he has done. He has said in one of his paper, he has described that 100% sensitivity through these NIPS because of low basal metabolic index in Indian women, because they are not obese. Yeah. But if you compare with Western and other countries, they are quite obese. Oh. So this happens due to low fetal fraction. Yeah. This false positive. Insufficient fetal DNA. Yeah. In the maternal bloodstream. Yeah. Or if the abnormality is mosaic. Yeah. If you have two different cell lines, with only small portion of cells displaying the abnormality. Yeah. So this is one aspect, is false negative. Yes. When I talk about false positive, results occur when NIPS, non-invasive prenatal screening, indicates the chromosomal abnormality that is not present in the fetus. Yeah. So factors contributing to this include technical errors during sample processing or analysis, maternal mosaicism. Maternal mosaicism will be there. Maternal chromosomal abnormality is also there. And there will be vanishing twin syndromes in addition to some translocation, which you cannot pick up. Correct. So these are actually the condition affecting DNA in bloodstream. Right. So I think this is a very good information that you have shared with us today. And it was great to have you, Dr. Bibhas, with us. Thank you so much. Thank you, Dr. Manisa. Thank you.